everyone, this is the second video of this Unit 1 playlist. In this video, we'll be explaining lipids and proteins without hemoglobin and collagen. Or with it, we'll see. Okay. Lipids. Lipids are another group of organic molecules consisting of carbon, hydrogen, and oxygen only. Lipids have many roles: energy storage molecule, part of cell membrane, thermal insulation, supporting and cushioning organs, and buoyancy. There are many examples of lipids: triglycerides, phospholipids, etc. The roles should be memorized, and we will be memorizing them together in the important video. We'll be named this. This chapter only studies triglyceride lipids. Later on, we will be doing phospholipids. Triglycerides consist of two molecules, glycerol and fatty acids. Triglycerides are formed when one molecule of glycerol combines with three fatty acids called tri-3 with three fatty acids in condensation reactions with the removal of three water molecules, forming ester bonds. This condensation is known as esterification. Quick note, it's better for you to watch the other videos because we've explained biological molecules separately. We've explained the condensation reaction separately. We've explained ester bonds. That the other one is called stuff, So yeah, this one will also be in the drawings video. The properties and type of glyceride form, known as either fat or oil, depends on the type of fatty acid. Because glycerol is the same in all triglycerides, fatty acids vary in two ways: carbon chain length, saturated or unsaturated. If unsaturated, then mono or polyunsaturated. Let me tell you something. Now, the fatty acids can either be saturated or unsaturated, right? This is one. These are two, which makes it recognizable, right? First of all, what we need to know about the saturated, it's solid, such as butter, and here it's liquid on room temperature, because this is important, such as olive oil. From the word olive oil, we must know, remember that the unsaturated comes from plants. So you can remember it by the olive oil, okay? And that the butter comes from animals. We've also talked here that this is 1, this is 2. Here, in the saturated, it's carbon, and in the unsaturated, carbon, double bond carbon. So 2 and 1. Okay. Saturated is solid at room temperature, such as butter, and the butter goes to animals. Unsaturated is liquid at the room temperature, such as olive oil. Olive makes it plants. Carbon, double bond carbon. 2, 1, carbon, 1, carbon. Okay. Exam questions. Saturated and unsaturated. Similarities: both have carboxylic acid group, both have hydrocarbon chains formed from only hydrogen and carbon. Differences: saturated fatty acids have no carbon to carbon double bonds, only have carbon to carbon single bond. Saturated fatty acids are straight chains, and unsaturated fatty acids have bent chains. Why unsaturated and saturated have different melting temperature? The greater the number of double bonds, the lower the melting temperature, because the fatty acid chain is more bent. So fatty acids do not pack closely together. Weaker intermolecular forces, less energy needed to separate fatty acid chain molecules. This is why animal fats are solids, while plant oils are liquids. But we also still do the exam questions in the other important video. Proteins. Proteins are organic macromolecules. We've talked about the macromolecule, and that all polymers are macromolecules, but not all macromolecules are polymers. Why? Because here is the example that I always draw. You must know that a polymer is made, for example, of a square, and it's this length. So this is a polymer, and so it's big, and it's a macromolecule. So they're made of same units. But for example, a normal macromolecule, which is not a polymer, is basically when you put a triangle, square, circle. So basically they're still big, so it's macromolecule, but they're not the same, so it's not polymer. So basically, pardon, just read this part. Basically all polymers are macromolecules, but not all macromolecules are polymers. Okay. Proteins. As I said, they all are organic molecules containing CHO and other elements such as phosphorus and sometimes sulfur as well. Proteins are made up of amino acids joined by peptide bonds by condensation reaction. So as we know here, the monomer is amino acid and it's joined by condensation reaction forming peptide bonds. That's why proteins are called polypeptides. Amino acids are monomers of proteins. There are only 20 types of amino acids, among which 12 are made in body, non-essential amino acids, and rest 8 are made from diet, essential amino acids. By using 20 in different combinations, different proteins are made. Amino acids consist of a central carbon atom joined to a carboxylic group at one end and an amino group at the other end, joined to form joined to a hydrogen atom and a R group. Our group varies among amino acids. This is important to know, and we'll be explaining it later as well, memorizing it later together, but it's important to know, and it has lots of questions about it. I'm gonna also put a YouTube video inshallah on the Telegram channel, Igcse.science.notesz. Try to watch it, it's gonna make it easier for you. How amino acids it's not my video yani, so I cannot say it now. Okay, it's from another channel called Crash Course. How amino acids join to form a polypeptide chain. The carboxylic acid group of one amino acid reacts with the amino group of another amino acid in a condensation reaction. Condensation basically is the reaction that bonds, and the hydrolysis is the reaction that breaks. In a condensation reaction with the removal of one water molecule forming a peptide bond, a dipeptide is formed and eventually polypeptide. Now there are levels of protein structure: primary, secondary, tertiary, and quaternary. Proteins do not exist as simple straight polypeptide chains. Rather, proteins undergo further reactions between the NH and the C double bond O, and between R groups of different amino acids to exist as 4 levels of structure. The primary structure, those structures are very important to memorize. Primary structure, it's the linear and specific sequence of amino acids in a polypeptide chain, joined by peptide bonds. Secondary structure, it's the folding of the primary structure into a helix and B-pleated sheets due to formation of hydrogen bonds between the NHS positive and the negative C equal the OS negative of amino acids. Alpha helix is the regular repeated spiral structure of the peptide backbone. Beta pleated sheet is the regular repeated pleated zigzag structure of the peptide backbone. Tertiary structure, it's the further folding of the secondary structure to form 3D irregular shaped structure maintained by ionic bond, disulfide bond, hydrogen bonds formed due to interactions between R groups and amino acids from different parts of the peptide chain. Quaternary structure, it's the 3D arrangement of 2 or more same or different polypeptide chains due to interactions between R groups from the different polypeptide chains forming ionic disulfide or hydrogen bonds. Note that the disulfide bonds are only formed between amino acids with sulfur containing R groups. Cysteine and methionine are 2 such amino acids. Disulfide bonds can form between cysteine and methionine, but not between cysteine and methionine. Strength order, disulfide is more strength than ionic, and ionic is more strength than hydrogen, so hydrogen is the least strength one. There are types of proteins: globular, fibrous, and conjugated. Our syllabi only focus on globular and fibrous. The globular is basically spherical folded shape to give their name. They have complex tertiary and quaternary structures. They are involved in metabolic roles, for example enzymes. They are soluble in water, as they have hydrophilic R groups outside and hydrophobic R groups inside. They also have specific 3D shape to carry out their specific metabolic roles, such as hemoglobin. We're going to talk about the hemoglobin shape later. Fibrous proteins are long polypeptide chains that lie parallel, so are arranged into fibers by crossing links. They have little or no tertiary structures. They are involved in structural roles of the body. For example, they form nails, skin, hair, so they are insoluble water, as they have hydrophobic R groups on their outside. There are long polypeptide chains, and many such chains are linked to be very structurally tough. For example, collagen. Conjugated proteins are proteins that are not conjugated to other non-protein molecules called prosthetic groups. For example, hemoglobin, conjugated to iron, lipoproteins, to lipid, proteins to carbohydrate, and these affect the properties of the proteins. For example, glycoprotein are used as lubricants in mucus OH groups, and carbohydrate part of glycoprotein allow it to hold water, which makes them slippery and viscous, reducing friction. Now, last thing is basically the hemoglobin vs collagen. Hemoglobin is a globular protein, and as you know, basically, let me not this tab, this one. No, sorry, it's this. Hemoglobin from this part here. Hemoglobin is a water-soluble globular protein which consists of 2 beta polypeptide chains, 2 alpha polypeptide chains, and 4 hemoglobin groups. It carries oxygen in the blood as oxygen combined to the hemoglobin Fe2 positive heme group, and oxygen is then released when required. Hemoglobin is made of 4 polypeptide chains as well as 4 prosthetic groups, so it has a quaternary structure. Also revising it from this PDF because it's lovely. Not this one, the green one. Yes. Okay, here. Okay, great. Let's now go back to the place where we came from. Okay, that's the thing I was changing. Hemoglobin is a globular protein. It has a quaternary structure of 4 polypeptide chains, each chain conjugated to an iron-containing heme group. Hemoglobin has hydrophilic R groups on the outside, making it soluble and dissolved in water, as it forms H bonds with water. Hemoglobin has a non-repetitive amino acid sequence. R group interaction results in different bonds that hold hemoglobin tertiary and quaternary structure together. Collagen is a fibrous protein. It has a triple helix of 3 polypeptide chains joined together by hydrogen bonds. It has a repetitive amino acid sequence of hydroxyproline, proline, and glycine. Every third amino acid is glycine. It has hydrophobic R groups on outside, making it insoluble in water. Anything I basically screenshot is a lifesaver, and we're gonna make a separate video about that. We're gonna really talk about it in details and study it together and stuff. Now we have to know the compare and contrast the molecular structure of globular and fibers. This is very important to memorize, and as I said, we're gonna talk about it later. Have a nice, beautiful day. Bye bye.